This video is on the immune system. So in this video, we're going to go over quite a bit, really. But our topics are antibodies, B cells, T cells, macrophages, interleukin 1 and 2, MHC 1 and 2, CD4 cells, and phagocytes. So that's a lot. Let's see what we got. An antibody, also known as an immunoglobulin, is a large Y-shaped protein produced mainly by plasma cells that is used by the immune system to neutralize pathogens, such as pathogenic bacteria and viruses. The antibody recognizes a unique molecule of the pathogen called an antigen. Okay, and then we have these different types of antibodies. So the first one we're looking at is immunoglobin G, which will be symbolized by a capital I, lowercase g, and then big G. Immunoglobin G provides the majority of antibody-based immunity against invading pathogens, representing approximately 75% of antibodies in humans. Immunoglobin G is the most common type of antibody found in the circulation. Immunoglobin G molecules are created and released by plasma B cells. Each immunoglobin G has two antigen binding sites. Immunoglobin A, which is symbolized as capital I, lowercase g, capital A, is an antibody that plays a crucial role in the immune function of mucous membranes. The amount of immunoglobin A produced in association with mucosal membranes is greater than all other types of antibodies combined. Immunoglobin D functions mainly as an antigen receptor on B cells that have not been exposed to antigens. In B cells, immunoglobin D's function is to signal the B cells to be activated. By being activated, B cells are ready to take part in the defense of the body in the immune system. During B cell differentiation, immunoglobin M is the exclusive isotope expressed by immature B cells. Immunoglobin D starts to be expressed when the B cell exits the bone marrow to populate peripheral lymphoid tissues. When a B cell reaches its mature state, it co-expresses both immunoglobin M and immunoglobin D. And then the last immunoglobin we have is E. Immunoglobin E binds to allergens and triggers a histamine release. If you have an allergy, your immune system overreacts to an allergen by producing antibodies called immunoglobin E. These antibodies travel to cells that release chemicals, causing an allergic reaction. B cells, also known as B lymphocytes, are a type of white blood cell. They function in the humoral immunity component of the adaptive immune system by secreting antibodies. Additionally, B cells present antigen and secrete cytokines. B cells, unlike the other two classes of lymphocytes, T cells and natural killer cells, express B cell receptors on their cell membrane. B cell receptors allow the B cell to bind a specific antigen against which it will initiate an antibody response. Okay, so with T cells we have the cytotoxic T cells and the helper T cells. So you need to be familiar with each of those. A cytotoxic T cell is a T lymphocyte, which is a type of white blood cell that kills cancer cells, cells that are infected, particularly if they have viruses, or cells that are damaged by pathogens in other ways. Cytotoxic T cells are stimulated by antigens and helper T cells. Additionally, cytotoxic T cells kill body cells infected with pathogens by releasing perforin, which ruptures microbial membranes. On the other hand, helper T cells do not directly kill infected cells as cytotoxic T cells do. Instead, they help activate cytotoxic T cells and macrophages to attack infected cells or they stimulate B cells to secrete antibodies. Helper T cells become activated by interacting with antigen-presenting cells such as macrophages. So let's look at those. Macrophages are important cells of the immune system that are formed in response to an infection or accumulating damaged or dead cells. Macrophages are large, specialized cells that recognize, engulf, and destroy target cells. They are a type of white blood cells that engulf and digest cellular debris, foreign substances, microbes, cancer cells, and anything else that does not have the type of protein specific to healthy body cells on its surface in a process called phagocytosis. We will get to that later in this video. Looking at interleukin 1 and 2. Interleukin is any of a group of naturally occurring proteins that mediate communication between cells. Interleukins regulate cell growth, differentiation, and motility. They are particularly important in stimulating immune responses, such as inflammation. Interleukins are a subset of a larger group of cellular messenger molecules called cytokines, which are modulators of cellular behavior. Like other cytokines, interleukins are not stored within cells, but are instead secreted rapidly and briefly in response to a stimulus, such as an infectious agent. Once an interleukin has been produced, it travels to its target cell and binds to it via a receptor molecule on the cell's surface. This interaction triggers a cascade of signals within the target cell that ultimately alter the cell's behavior. Interleukin 1 activates helper T cells to produce interleukin 2. Interleukin 2 stimulates B cells to become active antibody-secreting plasma cells. Interleukin 1 and interleukin 2 are both cytokines, as we've said. Interleukin 1 is secreted by a macrophage and has antigens on its surface. It activates helper T cells to produce interleukin 2. Interleukin 2 stimulates B cells to become an active antibody-secreting plasma cells. It is released by activated helper T cells. Okay. Now we have MHC1 and MHC2. So MHC stands for the major histocompatibility complex. Easier just to say MHC. It's a set of cell surface proteins essential for the acquired immune system to recognize foreign molecules in vertebrates, which in turn determines histocompatibility. The main function of MHC molecules is to bind to antigens derived from pathogens and display them on the cell surface for recognition by the appropriate T cells. MHC molecules mediate interactions of leukocytes, also called white blood cells, which are immune cells, with other leukocytes or with body cells. The MHC determines compatibility of donors for organ transplant, as well as one's susceptibility to an autoimmune disease via cross-reacting immunization. The human MHC is also called the HLA complex. The MHC in mice is called the H2 complex or H2. MHC1 are very common molecules that bind to cytotoxic cells. MHC2 are molecules that bind to helper T cells. MHC2 molecules are found on macrophages, B cells, and activated T cells. MHC1 molecules, on the other hand, are found on almost every body cell. When looking at active and passive immunity, you need to know the difference. Passive immunity is temporary and involves antibodies being transferred from one animal to another. Nursing is a common example. Active immunity, on the other hand, is permanent. Recovering from being sick or receiving a vaccination are examples of active immunity. The CD4 count is like a snapshot of how well your immune system is functioning. CD4 cells are white blood cells that fight infection. The more you have, the better. These are the cells that the HIV virus kills. As HIV infection progresses, the number of these cells declines. When the CD4 count drops below 200 due to advanced HIV disease, a person is diagnosed with AIDS. A normal range for CD4 cells is about 500 to 1500. And finally, we have phagocytes. A phagocyte is a type of cell that has the ability to ingest foreign particles such as bacteria. It engulfs foreign bodies by extending its cytoplasm into pseudopods, surrounding the foreign particle and forming a vacuole. In the blood, two types of white blood cells, neutrophilic leukocytes, which are macrophages, and monocytes, which are macrophages, are both phagocytic. Neutrophils are small granular leukocytes that quickly appear at the site of a wound and ingest bacteria. Monocytes are larger with a large kidney-shaped nucleus.